These are biggies. We're going to spend a whole big unit on cellular respiration, another big unit on photosynthesis, and these are the names for the chemistry reactions that happen inside of these cell parts if we are in a eukaryotic cell. Because remember, these organisms, just like Golgi and ER, are membrane bound. You don't find them in prokaryotic cells. Bacteria, prokaryotic cells. So here we have those, right? So we talked about ATP. We've already mentioned that mitochondria are the place where they get recharged. Where's the energy from that come from? The breakdown, catabolism of mostly carbohydrates and fats. Okay, what happens inside chloroplasts? Photosynthesis. And again, only eukaryotic cells have these two. So this is a big conceptual process kind of thing that we humans have figured out. It appears very clear from this evidence that these two cell parts, both of them, had as ancestors prokaryotic cells, bacteria cells, that got engulfed by bigger prokaryotic cells, and instead of getting digested, which most engulfed things do, they stayed alive, and as time went by, lots of time, they evolved to specialize in respiration and photosynthesis and to still live inside of these cells. Even though they can no longer live by themselves, they're too specialized. There are things about them which make it almost certain that this is where they originated long, long ago. Those things that make us think that basically number 3 things. They both have double membranes. You would get a double membrane if you got engulfed by a cell and you got surrounded by its own cell membrane. They have chromosomes and ribosomes, just like cells do, but they have the type that bacteria cells do. Even though they're now inside a eukaryotic cell, they have prokaryotic-like chromosomes and ribosomes. And then cells don't make new mitochondria and chloroplasts. They make themselves. They divide just like bacteria cells do. We call that binary fission, right? They divide just like that. So here's a good video to show that and also to differentiate some more of their structural parts. So now, almost all eukaryotic cells have mitochondria. We can pretty much say all, but there's almost always a couple little exceptions, right? They have an outer membrane, like we said, and then their inner membrane is folded. We call those folds cristae, just some more vocab, right? Notice these aren't in red, and you're not going to see them in red. They're not EOC things, but they are going to be on your tests in class, so that's why I'm saying them, right? And then inside that inner membrane, all the stuff that's in there is called the matrix. And this is where you'll find that bacterial DNA and those ribosomes. They didn't put them in this diagram. They really should have, just to emphasize that, right? And so that's the basic structure of that mitochondria that we're going to come back and look a lot more specifically at the respiration reactions that occur in there to recharge ATPs. Plastid is a kind of cell part. Chloroplasts are one kind of plastid. They're the most common kind. They're the kind we'll talk about almost always. But there's other plastids that have different functions, and you can read them here, right? What makes a flower petal colored is the fact that its cells have a whole bunch of these kind of little plastids that store those kind of colored molecules we call pigments. Okay, so what a chloroplast does again is photosynthesis. And what that is taking carbon dioxide and water and making sugars and oxygen, if you remember. Okay, we'll get to that much more specifically later. Right now, just they have chlorophyll. That's what makes them green. and they do photosynthesis. So that's what you need to remember about chloroplasts. So here's their structure. Here's some more names of parts inside, just like we had matrix and cristae for mitochondria. We have stroma. That's kind of like the matrix of a chloroplast. If you want to make the analogy, it's the inside, the fluid and things is called the stroma. The inner membranes are folded into sac-like structures called thylakoids, and they're folded into stacks of those thylakoids that are called grana, a word that means literally stacks of coins because they look like stacks of coins. So try that one. They love this kind of question in EOC land. First of all, they're telling you a story, and they want to know if you can apply your knowledge to that story. Secondly, they have a model, a diagram with labels. They expect you to visually be able to identify these things, but they also expect you to know their function. And so that's what they ask you here. Basically, his problem was producing sufficient energy. They expect you to know that that's mitochondria's job, which organelle was the source of his malady, a mitochondria, but they don't give you that in the list. They need you also to know what mitochondria look like. So of these things, right, that's the nucleus, that's out, right? That's clearly Golgi, that's out. This would be, ooh, what would you say, smoothie R, yes. Here in this diagram, it's correctly positioned, like I told you before. The rough ER is closest to the nucleus, the Golgi's furthest away. This is a more accurate diagram. You have these things. Well, they don't look like mitochondria, but these things do. Mitochondria are always, even though they're not always shaped this way, they're always going to kind of look like a hot dog with some, and you can't see it too clearly here, with some squiggles inside to represent those folded intermembrane cristae. So that's B is the answer, right? But B is not the answer here because the thing that's labeled B, just to be confusing, I think they made choice D. You won't get those kind on the EOC test. At least I hope not. This is a neat picture. This is an actual cell, a micrograph, and it's taken with a very special kind of microscope and very special kinds of isotopes that glow, or actually proteins that are made with isotopes that glow. And what you see are these green stringy things that go all through the cell. They are part of a cell's skeleton. So we use that same word in a cell for what we have in our cells. Here you see an electron. This is a scanning electron microscope picture. You can see the surface of these inside of a cell. So this cell's been cracked open literally. And you see how extensive this stuff is. It looks like a very dense spider web. And this is all throughout cells. It's so thin and so small, though, that when you look like in our class under a microscope, you can't hopefully see these because they're way too small, but they're there. They, just like you, are what give a cell its structure. If it's an animal cell, its plasma membrane is way too flimsy, like your skin. If we took all your bones out, you would just collapse on the floor in a heap. Your skin is not strong enough to keep you, keep your structure, keep you upright. It's your inner skeleton that does. That's what the cytoskeleton is to a cell. Cyto is the cell prefix, right? So what your skeleton is also involved in is movement, right? In your skeleton, it's muscles attached to your bones that when the muscles contract, your bones move. So here is a little way. Now we're going to add to our story about things being made in a cell and transported outside. If you pretend that this was one of those vesicles that had enzymes in it made in your pancreas, and it's going to be sent out of the cell, the way that vesicle is going to move in a cell is that a little part of this cytoskeleton called a microtubule is going to be like a railroad track. And a little protein, in fact, all these are made of protein, a little protein molecule that when it's energized by ATP will change its shape and move, is going to grab this thing and move it along the railroad track. Very neat little molecular motors inside of your cells moving things around. So these two cell parts are like little hairs or like a tail. That's what they look like, right? Cilia are little hairs. They're usually multiple on a cell. A flagella is like a longer hair or tail. It's often singular. It might be multiple, but it's often singular. And they are going to help these cells move around. But inside of these cilia and flagella are microtubules. The difference between the two is really only their length. Inside, they're both the same. So we're going to watch a couple little videos here as I skip through these. And we'll see some cells that are being moved along by cilia and flagella with microtubules inside. And then we'll see some other kind of movement by the other kind of part that makes up the cytoskeleton. Excuse me. So that's a little one-celled thing that has cilia, and it's moving around with this cilia. You're also going to see contractile vacuole inside here. And the contractile vacuole, you're going to see it pump. It's the other kind of cell part that's causing it to change its shape and pump water out. And we'll talk about that when we get to it. See it moving around? Quite neat. You see that contractile vacuole? That thing just closed. That's being squeezed by the other kind of cytoskeleton part called microfilaments. We'll get to talk about them in just a second. But this thing and all those other little things are moving around by cilia, which you can't really see too well in this micrograph, but it's kind of neat to look at the micrograph. So now you have microfilaments, which are thinner, again protein, microtubules and filaments are all. This is the specific kind of protein that makes up most microfilaments, right? So these things cause things like your muscle cells to contract, these microfilaments. So this kind of movement we're talking about is a change of shape. Your muscle cells don't move around like that little creature was moving around in the pond water. They move by changing shape. And then sometimes inside a cell, as you'll see in just a second, the contents of the cell can get stirred around, kind of like stirring your chocolate milk, right? By these microfilaments causing things to move around and circulate things in a cell just like your blood system circulates things in you. So there's some of that with the music. I don't know if you can hear that and me. I haven't done these before where I'm playing while I'm talking, so I'm skipping right now. Okay, so I'm going to show you one more where the microfilaments, again, this time, are causing a cell to kind of ooze around and therefore move from place to place, but not because it's got little cilia on the outside like that other thing was moving around. This is microfilaments inside causing it to change its shape and send out a part of itself that somebody decided we'll call a false foot, pseudopod, right? And then it'll move the rest of itself towards that. This is called amoeboid movement because the cells that often do that are those kind of cells we call amoeba. There you see. Very neat to look at under a neat microscope. You see all that stuff inside a cell too. Very, very complex inside that one cell. And this is a one-celled organism, but it's got a nucleus, so this is eukaryotic, right? So there you go with that. Okay, this is like getting to the end here, which is good. Now we have cell walls. not to be confused with cell membranes. And it's easy to do that, right? Every cell has a plasma membrane, like I'm drawing, right? Or I'll draw it over here because my face might be over there, right? Every cell has a plasma membrane, plant cell, animal cell, prokaryotic cell, or whatever. Many cells have cell walls also. None of your cells, this is a big animal cell, plant cell difference, but all prokaryotic cells do, all plant cells do, all fungus cells do have cell walls. The cell wall is outside and much thicker than the plasma membrane. So if you're bumping into a plant cell, the first thing you bump into is its cell wall, and then once you get through that, its very thin plasma membrane is right under that, okay? Its position and its thickness suggests one of its functions is to protect the cell, to maintain its shape. Unlike flimsy animal cells, a plant cell gets its shape from its cell wall, not its cytoskeleton, even though it has cytoskeleton. Okay? And what we're going to see in the next unit, in much more detail, is how this cell wall also prevents this cell from taking in too much water. And then if you look at an entire plant made of cells with strong cell walls, that's why that plant can stand up and compete for sunlight. So just a little more detail. This is outside the EOC syllabus stuff about cell walls. Cell walls come in two layers. The layer that's made first by a plant cell is the primary cell wall. When it's a little baby plant cell, it makes all these molecules and secretes them outside. And then secondly, it makes a cell wall that's much thicker than that primary cell wall. And in between the two is a part called the middle lamella, which is a word that means layer. So that literally means middle layer. The cell walls of plant cells, which we're focusing on here, is made mostly of polysaccharides. And specifically for plants, that polysaccharide we call cellulose. There's a couple others. There's some protein stuck in there too, but cellulose is the main component of plant cell walls. It also holds the distinction of being the most abundant organic molecule of all of them. Of all the different kinds of organic molecules, there's more cellulose molecules than there are any other kind. Just trivia fact. So here's a couple other molecules that are found in cell walls. They help hold the cellulose together. If you remember from when we talked about cellulose, cellulose is a long string of glucoses. They're all spiraled up and they're all banded together to make real-like tough ropes. What those ropes, those fibers are held together by are some sticky ones called pectin and lignin. And it's actually pectin, for instance, that makes apple pie sticky. Pectin is a sticky molecule, and when you boil apples up to make the filling for an apple pie, the pectin comes out. It then solidifies when you cool it down. Pectin is also what makes jelly jelly-like. So I'll let you practice with these, right? But these are very classic test questions in the compare and contrast mode. We have differences and similarities, right? Similarity, compare, difference, contrast between plant and animal cells, prokaryotic and eukaryotic. Mitochondria and chloroplasts have similarities and differences. So good practice is making Venn diagrams about those three different similarities and differences. So here's another good practice. We have told the story of a cell like a pancreas cell making some enzymes and sending them out. Which of the cell parts that we've talked about are part of that story? And what is the specific function of each of those cell parts? We'll do this in class, but you can practice here with this video, which I think is over or close to over. There, it's over. This is, I took this about a week ago. Sunrise, Jack's Beach. Hope you enjoyed that one. Took me long to make this one. Bye.